My name is Ken Janda. I'm lucky enough to be the Dean of the School of Physical Sciences. It's my honor today to talk to you for a few minutes and introduce our speaker. A couple of announcements. First of all, thank you so much to the people who have supported our school so well and helped make this breakfast series happen. I'm going to have a special announcement in just a moment, but we'll first talk about some of the good news about UC Irvine. So one always reads about how universities are, for some reason, the popular press seems to think we're losing our game. Well, UC Irvine has the highest growth in freshman applications of the whole University of California system, including from underrepresented minorities and local residents. So at least the people who really matter in our business model, if you'll forgive that word, are showing great confidence in the university, and we're really proud of that. Some of you may have heard that we're trying to get President Obama to speak at commencement this year, and that would be pretty exciting. It would happen to be the 50th anniversary of when President Johnson came here to dedicate the land for the university. And today we're proud of all of our supporters, but especially proud to be joined by Don Beal this morning. Some of you may have been at a wonderful performance last night at the School of the Arts. We're celebrating the Beal family's support of the Arts and Technology program at the School of the Arts. The Beal family has also been a huge supporter of the School of Physical Sciences. One of Don's passions is bringing intellectual property to the public. And this year he's supporting 3 of our faculty members' research and helping them bring it closer to fruition and bring it to the public. And some of you may have noticed, was it last week, an announcement of the new establishment of the UC Irvine Institute for Innovation, and that was largely motivated by Don's work and his influence in the university. So we're so proud that Don is also a supporter of our school in addition to the rest of UC Irvine. And thanks for coming this morning especially. So other things going on. Oh, Mark, welcome. Other things going on in the school. You may have noticed that it's been kind of dry in California with the exception of the weekend 10 days ago. So the Professor Jay Familietti from the Earth System Science Department has joined the State Water Board to advise Governor Brown, and he's also working with Barbara Boxer on helping her understand those issues better. One of the interesting things that happened in chemistry recently is that there are several divisions of the American Chemical Society, and one of those is the Division of Physical Chemistry. They give 4 national awards every year, and 3 of those came to chemists here at the University of California, Irvine. Something that's a little more local, the local math counts competition had winning teams from Jeffrey Trail Middle School, San Joaquin, and Sierra Vista Middle Schools, and those go on to the state competition on March 15th. That's also here at UC Irvine, right? And Jeffrey Trail Middle School, in addition to winning the math counts, won the Orange County Science Olympiad competition, and they'll be going on to the state competition. And our students helped train them for that, so we're very, very proud of that. Today we're focusing on the physics department, so a couple announcements from physics. Professor Tim Tate has been elected as a 2013 American Physical Society Fellow for his exceptional contributions. And there's a show going on right now at the University Center called Particle Fever, which talks about the Higgs boson project that our Professor Andy Langford has been very influential in, along with half a dozen other UCI faculty members. So see that. Do we know if it is extending past Thursday, or do you have to see it by Thursday?
I don't know.
Yeah, so anyway, a chance to learn about particle physics over at the University Center. And then a really exciting development from the Department of Physics and Astronomy, the Simons Foundation has given out a $2.5 million project, part of which, about a quarter of which, is located here at UC Irvine under the direction of Professor Steve White. So this project is to understand the quantum mechanics of electrons, which determine all the chemical properties of just about everything we come in contact with. And that's one of the hardest problems in chemical physics. And so we're very proud to be a center for that. So once again, thank you for all of our supporters. If you'd like to join the group, there's pledge cards on the table. Big surprise. But seriously, it really does. Every penny I think we spend very carefully, and it really helps our students go on to greatness. So today it's my distinct honor to introduce Professor Zuzanna Siwy. Zuzanna was born in Poland, where she earned her Ph.D. from the Silesian Technical University of Dubisa. Is that even close? From 2000 to 2003, she was a fellow for the Foley Science and Alexander von Humboldt Foundation. After completing her postdoctoral studies at the University of Florida at Gainesville, she joined the faculty here at UC Irvine, where she quickly went on to fame and fortune. She's been named an Alfred E. Sloan Fellow, a National Science Foundation awardee for the Presidential Early Career Award, and she was selected for the top tier of those awards, being invited to the White House to receive special recognition, and just recently was elected to be a fellow of the American Physical Society. So Zuzanna is going to talk to us about her work in physics, biophysics, and I understand the title is Cancer, Anthrax, Flu, Water Desalination. What is the link? Zuzanna.
Thank you, Ken. Thank you, Ken, for this really, really nice and generous introduction. And let me start with saying how honored I am to be here with you and to share with you some of the topics which my research group is excited about and some of the questions which we try to answer. So every time we open a newspaper or we open a browser with news, apart from good news from UCI, it's normally bad news, okay? There's a lot of worries which we see all the time. In my opinion, there is not enough good news in the news. And certainly, one of the worries which we are constantly reminded about is the worry about cancer. Of course, we live in an amazing place where we have a few, or more than few, extremely good choices to find hope and cure. But I think you would agree that it would be really useful to have even earlier methods of detecting cancer, and certainly more personalized medicine. We know that one course of treatment, which is very effective for one person, unfortunately does not have to be so for someone else. Another reason of worry, which Ken already mentioned about, is the drought which we have in our state. We worry about a lot of things. We worry about higher prices of veggies and fruit. And if you think about it, it is a little bizarre. We worry about water because we live close to Pacific. Pacific is an amazing reservoir of water. Unfortunately, not really viable for drinking water or farmland because of its salt. And desalination currently is available, but it's very, very expensive and it's very, very inefficient. So we really do not treat it as something which would be useful for that. And eventually, that's a picture of bacteria, Bacillus anthracis, which carries anthrax. And of course, we still remember the very sad news we got over 10 years ago about bioterror attacks with anthrax being one of them. So during the course of the next half an hour or so, I will tell you a story. It's not really a presentation, it's a story about how physicists approach those very important issues and how looking at biology, especially at how biological cells function and communicate with each other, can lead to new solutions and approaches. I will be referring to cells, sometimes part of cells, as devices, okay? Devices which can do something extremely useful. So kidneys are this amazing desalination unit. Our nerve cells can be thought as wires connecting different parts of our body, and eventually I will talk about our ear, which is our amazing amplifier. So I'm switching gears a little bit, and when I learned one thing which I think surprised me most, if not shocked most, when I started to work in biophysics and physics and learning about biology was that lots of diseases, including invasive cancer, Alzheimer's, neurological disorders, depend on proper functioning of tiniest structures you could imagine, which exist in every part of our body called pores and channels. And they will be the subject of the first part of the story. Another thing which surprised me is that we would not be able to hear or see, or I wouldn't be able to move my hands, again, if not for the existence of these beautiful structures which are embedded in every cell. Now, from the technological standpoint, desalination is so expensive because the pores which make a decision or make separation of water and salt possible are not optimized. That is why it's so expensive. And I could definitely think about the new blood tests for cancer, for viruses, would also be based on these beautiful structures which are called channels and pores. So what is a nanopore, and how is it possible that so many important processes depend on them? So we all know what a tunnel is. You might be like me, a little anxious to enter a tunnel. I don't know when the exit is. I'm a little claustrophobic. But anyways, through big tunnel, we can have big things passing through trains and cars and bikes. If you make the tunnel a little smaller, like one in my favorite mountains in Poland, a toddler can pass through easily, but an adult has to kind of fold in half to get there. Now, if you make the tunnel even smaller, comparable to the size of hair, that's about a micron or tens of microns, and now you make the hair about 1,000, if not 100 times smaller, you will get a system which we'll be talking about. So it's the tiniest possible tunnel. It's so tiny that you can look at the passage of individual molecules or individual ions passing through. But biology operates in water, okay? 70% of us is water. So we really should think about channels which are filled with water. So think about this very tiny structure which is separating two big reservoirs of water on both sides. Okay, so where are the nanopores placed in our body? They are everywhere. Every cell is surrounded from the outside world by a membrane. The membrane is a barrier. It's very fatty. Almost nothing passes through. And then, but the cell sometimes has to exchange nutrients, has to exchange ions and molecules. So we have those pores and channels which are embedded in it and connected by as a whole, basically, connecting inside of the cell with outside of the cell. I call it smart hole because a lot of them are selective, so they allow only one particular molecule or one particular ion to pass through, and they are extremely efficient. Every second of our life, if the pore is open, allows millions of ions passing through each individual channel. It's amazing. And they are also responsive. So many of them do not do anything unless a stimulus comes from outside and does something. So one of the stimuli, which is very important, is sound. Okay, so sound wave comes in to our ear, and then through ear canal and eardrum and those tiny little bones, it's transferred to cochlea. And cochlea has a snail-like structure. If you open it up and you make it planar, you will see a V-shape things on it. And if you zoom into it, you will notice that each of the V letter-like things consists of many cells called hair cells. And they are called hair cells because what is sticking out, what you can see, they are hairy things which are very flexible, which can move. So when the sound waves comes in, the hair is actually deflecting, is literally mechanically deflecting as the sound waves comes in. And these little hairs are connected with one another via a linker, which opens a channel. It's a mechanical pulling on an opening of the channel. It's a very quick process. What happens is the ions which are here are entering to the hair cells, and they are detected by the brain. So that's the electrical signal which is detected by the brain, which means that even though all the other components of the ear would be functioning, but the cells would be dead, we are deaf, okay? So this is the absolute linkage of the sound waves to the brain. It's really amazing. They are very precious, and they are actually very few of them. We have only 20,000 of so-called outer hair cells and 17,000 of inner hair cells. It seems like a lot, but if you compare it with millions of cells in our eye, it's very little. It's very hard to study them because they really do not like being exposed to vibrations. So the channels are absolutely crucial in the hearing part. What was discovered fairly recently is that some of the cells, called outer hair cells, they do not only detect the sound, but they amplify it. So what happens is the sound comes in, the channel over here open up, the signal is transmitted, but they start vibrating, they start changing their length. And that changing of the length, which you will see in a moment, actually is enhancing those mechanical vibrations that are coming out from the mechanical wave. So I want to show you one of the most popular videos on YouTube, which shows the movement of the cell. So that's the vibration. Well, I guess that the cell got tired of it. Let's see. Okay. You see? Oh, I know what I'm doing. It is me. It is me playing with the remote control. I'm not used to it. That's the reason. Nothing is more annoying than someone trying to show you a video which doesn't work. So it's literally moving, and that mechanical linkage. Okay, I guess it's enough of rock and roll. So anyway, so we can really see it happen, and those vibrations cause amplify of the signal, and that is why we can hear so well. In fact, so our ear can detect the sound, but it can also produce it. And this is the way how now in infants and newborns we can check their hearing test. So that's the hearing test, which is based on production of the sound waves. A little explanation, I'm not trying to advertise for any particular hospital. It's just a picture of my daughter. So if you see a little hat with a particular name of the hospital, it's not supposed to be an advertisement of anything. Just a little clause. Another place where biological channels are absolutely crucial is the nerve cells. Okay, so I would not be able to move my hand, which I'm doing probably too much without it. So nerve cells can be thought about as wires of our body. They connect one part of the body, one cell with another cell, and they relay information. So I'm going to show you how that actually works. And this is one of neuron cells. A neuron cell has this very long part called axon, which can be very long. Okay, by very long I mean some of them are 3 feet long. That's long. Okay. And I call them transmission lines because their main purpose is to transmit signal from the stimulus which comes in to the nerve cell. And the important thing is that the signal, and I will tell you what the signal is, has to be transmitted without any losses. So if the length is 3 feet, at the end of the 3 feet, the signal has to be the same as in the beginning of the 3 feet. So if you magnify it, you will see a cylinder. That's part of the axon with a bunch of embedded channels. And one of the 2 key players is a channel which is selected for sodium, which opens up when a stimulus comes in. And then that opening of this channel and influx of sodium causes another channel to open up, which is potassium. Again, imagine thousands of ions passing every second through them. It's amazing. And then if you took your multimeter from home and stuck it inside and outside of the axon, what you would see is a pulse. is a pulse of a physical property called the voltage. The voltage change would have a shape of a pulse, and that's so-called action potential. So the electrical signal is actually what is transferred by our nerve cells. It's truly electrical. So now what happens is that the neighboring channels feel the pulse coming in. So again, the situation repeats. The sodium channels open up, then the potassium channels open up, and now repulse in a new location. So the reason why the signals are transmitted without any losses is because they are constantly being regenerated at new locations. And it happens at a speed of about 200 miles per hour. If I'm sleep deprived like now, I'm probably transferring my nerve signals a little slower, but I guess 200 miles per hour is a good estimate of what our body is able to do. And now we might wonder, and then what? The signal comes out. This is one nerve cell. And now we have to relay the signal to another cell. And that happens by a coupling of electrical and chemical signal. So there are little molecules get released called the neurotransmitters. They have to get out of the cell. They are wandering around in this little space between two cells. And they come across another type of channels, which will be important for the next part of the talk. And these channels are zoomed in here. They are closed. They don't let anything to pass through unless two of the molecules of neurotransmitter molecules come in and bind very specifically, and the channel opens up. By the way, those channels are also target of many toxins. Okay, many plant toxins and snake toxins, they are so bad for us because they mimic or they cheat us. And instead of the neurotransmitter, which is good for us, a toxin binds to it and the nerve signal cannot be transmitted. So pharmaceutical companies are also thinking about, or are also making drugs which would affect the ligand-gated channels. So ligand-gated channels are, again, is the inside of the new cell, outside of the new cell. The signal gets transmitted, ions are getting in, and again, the cell knows that there is a signal coming up. Another pore, which made a lot of news recently, a lot, is so-called aquaporin, which is a pore for water. There was another prize for it 11 years ago for Peter Ager. And aquaporin is the structure which allows transport of water with an amazing efficiency. And what I mean by that, it's 1,000 times more efficient than any currently designed desalination unit. So what I'm showing here is our little desalination station, which we have in our lab, which we use every day for preparation of solutions. It's very, very slow. Sometimes people ask me, it looks very messy. I agree, sometimes this looks a little like thrown together with someone, but actually it's not. It's a very well-designed protein structure, and the water passes through one by one. It allows only water to pass through. All the ions are staying outside. The perfect desalination unit ever created. They are all over our kidneys and in our parts as well. So anyways, of course, the military is very interested in it because if you could create now synthetic, by that I mean man-made pores of similar efficiency and performance as aquaporin, you could drastically improve performance of currently existing desalination units. So if troops are sent places without easy access to water, they are often accompanied by lots of trucks loaded with desalination unit. If the desalination units had aquaporin-like pores, they would be able to reduce the size of the backpack. Of course, the size of a military backpack is a little larger than the size of our backpack. Nevertheless, it would be much smaller than a truck. So we actually got a little money from DARPA to do that with our nanopores to increase efficiency of desalination units currently existing. But you might wonder what I'm doing here, being from physics department, right, talking about biology, it seems like a little disconnect. So I will tell you what my colleagues are excited about when I tell them about biological channels. And what they tell me is that they are fascinating because they really behave like devices, okay? So a lot of them behave like devices which they know from electronics. So they behave like diodes or they behave like transistors. So a pore behaves like a diode because it transports ions only in one direction and blocks it in the other. For example, a pore would transport ions from inside of the cell to outside of the cell, but it would not allow transport from what did I say? Okay, yeah. to inside to outside, but not from outside to inside, okay? That's a switch, perfect switch. Some pores are like transistors, meaning they can, in some ways, amplify the signal. So there's a tiny little change, really little, happening next to them, but they respond hugely, okay? So the transport properties are humongous. So now let's think about it. You know, we love putting diodes, transistors, capacitors in integrated circuits, right? This is what it's all about. In every smart computer, in every smartphone computer, we have them. So what if we created nanopores which would behave like biological channels, do all these amazing properties, but we could combine them in a circuit. We could connect them all together in an integrated ionic circuit. I think we could totally revolutionize how we detect molecules, how we detect cells, how we detect viruses, because all these things have to be detected in water. Okay? Electronic devices do not like water. We cannot use them in a water solution. So we really have to have a different approach to do that. And I think biology gives us the best way of approaching it, because even in the axon potential, even in the axon part, we had many channels cooperating with each other to achieve a given function. So I think we really created a new field, and that's the prize of the President Obama, which we got for, because I think that there is something which we can do. Okay, so we need nanopores. Okay, we need small structures with very small openings, ideally in some cases comparable to openings of biological channels. So the whole thing starts, in our case, in Germany, where I spent I spent 3 years and I made amazing friendships, and it has been a wonderful time. And we are using their linear accelerator. It's about 300 feet long. The diameter is shown here. And then what they do, they take heavy elements, we like gold, we like uranium. We strip them out of many electrons, so they are ionized, and they accelerate it to amazing, amazing velocities. And what we do, we bombard our samples with them. So I brought little prompts, and there's a little test. Who can find a sample in here? There is one. Okay, so you are more than welcome to touch it, to open it, to break it. Okay, nothing can be. There are different shapes and different sizes, different materials. This one is yellow, that's Kapton. Okay, the transparent one is polymer. So this is the materials we are working with. Okay, rubber thin, and you're more than welcome to do whatever you want with test them. So they are irradiated, they are bombarded with these heavy ions. They go all the way through. Sometimes I give analogy, which is probably not quite accurate, but it's like shooting through a big glass of window. There's only one hole in it, and the window hopefully remains intact. So it's a similar thing with that happening. The ion is passing through. It locally damages the material. It makes it weaker. And those samples are sent to us, to Irvine, where we subject them to further processing. Also, whenever I talk about it, people say, oh gosh, you need accelerator, 300 feet, that must be really expensive. It is, but it is and it's not. I'm answering like my toddler a little bit. It's not, because if the process is optimized, you can irradiate 1,000 samples per hour. So the cost per sample is actually very small. So yes, you do require, in this particular case, a heavy machinery, but it is already in place, and it's not that expensive, in fact, to use it. So in Irvine, we perform wet chemical etching. So we put it, depending on the material, into bleach, into sodium hydroxide, into acids, and we etch pores. Okay, so we make real holes with the diameter and shape, which is tuned by the chemical etching. Depending on how the development of the holes is performed, you can get holes which are cylindrical in shape. Some of our favorite ones are cone shape, meaning that they are wide on one opening and very narrow on the other opening. We also made, we are very proud of it, we actually patented, UCI holds the patent of a technology to create very versatile pores in silicon nitride. That little chip over there is the silicon nitride chip which we are working with. Silicon nitride is convenient to use because ultimately you have to interface your ionic device with some sort of electronics things, and silicon nitride allows us to do that. But in any case, if you look at this tiny little structure over here, it's an hourglass. There is a little hole, and that little hole has size comparable to the size of biological channels. So what do we do with them? Okay, what do we do with the holes? So I'm going to give you a glimpse of a minute from our lab, and I hope not to mess up the video this time. So anyways, we have a lot of electronic equipment in the lab. We have some devices where we put the membrane, the hole between, this is a little better. That's one of the polymer foils we are using. We are clamping in between two reservoirs, so they are, it's actually shown over here. So we literally, it's very easy. It's done by our physical sciences machine shop. So we just clamp it together. It's very easy to handle it in hands. It's a very tough material which we are using. We prepare solutions, put it with a tiny little pipette into those holes, and we have electrodes and we measure current. By now, everything is nicely atomized. We can do it pretty efficiently on the fly. So one of the things which we did when I came to UCI was to try to make this ionic switch. Okay, was to try to make a system which would transport ions in one direction but not in the other direction like many of the biological channels do. And we were able to do that by very careful design of the pore shape, opening, and also what do we attach on the walls. Okay, so we decorated the walls with molecules which carry overall positive charge, and then we interface it with the zone which carries negative surface charge. And that very simple manipulation of the pores resulted in the first ionic diode, which showed very nice asymmetry. You can see the current is finite for positive voltages and it's 0 for negative voltages. So how does it work? The only thing which is important in this is that positive attracts negative. That's all what it's about. So if you work in a salt solution, you always have positive ions like potassium and negative ions like chloride. So the positive region attracts negative ions and the negative region attracts positive ions. You apply voltage, the ions are moving out. There is not much of them. So very, very quickly the current will cease, so there is nothing else passing through. But if you just revert the voltage polarity, which basically means that now the potassium goes not to the right, but goes to the left, the system transports ions beautifully. So the ion can be sourced through, and there's no problems in doing that. So it's one of the, I think, very nice examples of how important it is to manipulate size, geometry, and also surface properties. So the concept of ionic switch and the concept of ligand-gated channels I talked to you a little bit about before, we use that in preparation of a sensor for anthrax. Okay, so anthrax is carried by bacteria. It's Bacillus anthracis. It has the characteristic rod-like shape. and it can be observed with a light microscope. Okay, it's fairly large. What we realized, however, it would be useful to do is to be able to detect very early on if someone is infected with anthrax, okay, if you really carry it. So we learned from the literature that the bacterium is covered by so-called capsule, okay? I call it a coat, but it's actually a little capsule. It's built out of proteins and is actually built only one amino acid in case of Bacillus anthracis. But what is most important is that when the bacteria enter a host body, what happens is that the coat, the capsule, is shed very quickly, okay? And what they found is that those junks or the fragments of the capsule can be found in the blood within a day after infection. So one way of detecting infection would be to check for the presence of those coats of the bacteria. So we talked to Thomas Cazzo from the University of Nevada, who, by the way, is trying to develop a vaccine against anthrax. And he developed a molecule, so-called monoclonal antibody. It's a molecule which binds that capsule material very specifically and very strongly. And what we did with it, we took our pores and we, as we say, decorated, but really we attached the molecules to the walls, to the narrow opening. And first we check the sensor behavior before exposure to the coat. And the blue line represents measurements performed in conditions which are the closest to physiological conditions. And now, after exposing it to the coat from the bacteria, you see that the blue curve becomes 10 times larger. And it's somewhat similar to ligand-gated channels where binding of things from outside changes dramatically behavior. Even larger response was obtained at acidic conditions comparable to our stomach, where the change of the current was 100-fold. So we were happy to see a huge change of the behavior of the sensor, because a huge change of transport properties, it basically prevents you from false positives. But it would be unfair from me not to say that I have to tell you that pores have been used in medicine for a long time. Not nanopores like ours, but pores. So I want to show you as a glimpse of a patent application from 1953 from Wallace Coulter. And Wallace Coulter in 1953 proposed the simplest ever method of detecting cells and particles. And the method is based on a pore. The pore which he had at that time was larger than the thickness of our hair, but it was perfect for blood cells. 99% of the time, if you go to the doctor's office and they order blood tests and you see complete blood count, it's done by Coulter counter. So this is how it works. You have a pore, again in Coulter counter, much larger than the one I talked about. And you have, again, two reservoirs of liquid. Let's say that your blood is on the left-hand side. It's pushed through, so it's pumped through. There's pressure all the time. And then one cell gets in. Because the cell gets in, it partly blocks the pore. Okay, it occludes the pore. There's less volume in it. So you can see it as a change of the electrical signal. There's a drop in the signal through the pore. If you have a larger cell, the same thing happens, instead that now you have a larger signal. So it's the perfect method of checking the size of cells. Red blood cells are much smaller than white blood cells. That's the way how they differentiate them. You can find concentration. It's perfect. It's used by NASA for checking quality of fuel cells. It's used routinely by paint industry. You know that you pay a lot of money for a good paint because of the size of particles which are in there. So all that is based on Coulter counter. But Coulter counter has limitations. One of the limitations which the current technology has is that it's insensitive to shape. In many cases, it can be a plus. But in many cases, it's a limitation because a changed shape can be indicative of disease. And probably the best known case is the sickle anemia, where the red blood cells, rather than being round and spherical, they have the nice, well, actually not nice, but they have the sickle shape. Another physical property, as we say, which is very important, is we say it's Cushiness. And what I mean by Cushiness is how easy it is to deform an object, okay, how soft it is. So as it appears, malaria parasites, if someone has malaria, red blood cells became much stiffer, actually. And that's really bad for us because stiff red blood cells have difficulties in passing through efficiently very small blood vessels which we have in our body, and as such the oxygen flow is limited in us, and it's certainly bad news. But another reason why cell squishiness is, people are so interested in cell squishiness, because there has been a correlation found between squishiness of cancer cells and their ability to invade our organs, in other words, malignancy. And this is one of the cartoons which shows why the hypothesis actually makes sense. So what I'm showing is a scheme of a primary tumor somewhere located. And then at some point, it might not have to, a single or a few cells get detached from it. And when they get detached from it, they can, in principle, enter our bloodstream. It's not easy to enter our bloodstream. Okay, the blood vessels were very well designed, so they have to look through small cavities through which they have to squish through in order to enter the blood. So now the cells are in the blood. They are called circulating tumor cells. And now the scary part comes in. Being in the blood, they have access to the whole body. So in principle, they can come out. It's called extravasation, which is unimportant. It comes out, they might come out, and they might start growing secondary tumor somewhere else. It's a hypothesis. It's one of the hypotheses how people think malignancy can occur. So you can see that the squishiness here is very important because only cells which can deform, they can get in and out of the bloodstream. Circulating tumor cells have been a very hot topic, extremely hot topic. And one of the reasons, there are many reasons for it, but one of it is that the number of those circulating tumor cells has been correlated with the prognosis of a patient. So if a given course of treatment is working, then the number of the cells is decreasing, is dropping. Another way, if you could think about it, is that if you could harvest cancer cells from your blood, this is a non-invasive biopsy. Okay, you have access to it without any other very unpleasant ways of getting a sample. It's very, very hard to count them. There are only a few in a few milliliters of blood. There is one company, one device approved by FDA. It's very expensive. It's not widely used by medical doctors because it's not really that reliable for cancers. It's even harder to harvest them in a way that they are alive. If you could capture the cells in a state that they are alive, you could grow them outside in a dish. And you could test drugs on them before checking those drugs on the patient. So it's a lot of interest for personalized medicine in that. So we thought that we could design a method to capture them using physical methods, using physical approaches. Because physical approaches are typically, not always, but we can make them gentle. So we would capture them and separate them without damaging them. So we wanted to study squishiness. So how do you study squishiness? Well, you probably have to have a finger and just press on them and see, oh yeah, well they deformed, right? Like a Play-Doh. But you have to do it in a highly efficient manner, in a way in which you will not destroy them. So here physics came with help. So imagine that now your pore is not a cylinder. Now your pore it's not the final product, but it's narrower at the entrance, and it's wider in the middle. Okay, it has a wider cavity in the middle. And now we are pumping your liquid through, let's say, your blood. What happens is, in the zones where the pore is narrower, the fluid flow will flow faster. It's like if you have a river, and in the narrower part of the river, the velocity is faster than the wider part of the river. So here, the velocity of the fluid will be smaller. And if you have inhomogeneous, as we say, or if you have different velocities, you will get different pressures. And by calculating, you can actually design the pore so that the pressure drops which you get are sufficient to squish the object. So this is how it would work. You have a hard sphere at the entrance. This one doesn't deform. It passes through, it blocks the pore. Then it's here, it blocks the pore to a less extent, and then it comes out here. Now, if you have a squishy object, you start at the same, but then if the object really gets squished by the internal drops of the pressure, you will get a smaller change, and from the difference of it, you can quantify mechanical properties of each individual cell. This is our preliminary results. Laura in the audience did those experiments. So she mixed mice cells, which are potentially squishy, with hard spheres, which are not squishy, with particles. And those little ones are particles. They are 5 microns. The size was chosen on purpose. They are comparable to the size of our red blood cells. And you can see that they have that the signal upon entrance and the signal upon exit is pretty much the same. But the cell, you have a huge difference in the spikes, which might correspond to change of mechanical properties, which might indicate squishiness of the cells. So the project, actually, the idea, Laura, who is here right next to me, built a very successful team. They entered the business town competition last year. And believe it or not, they collected the largest check of $30,000 of the main life science award. And they were greedy, so they also got $35 additional for a third overall prize to hopefully bring it to realization at some point. So I'm almost done. I just want to make a few remarks of what we do and how we try to disseminate what we do. So basically, our daily life is busy with making holes. Okay, it might not sound very exciting, but that's what we do. So we make holes of different geometry, of different opening, with a hope that we can contribute to some of the very important issues which we have right now. Now, what we do is very interdisciplinary. It's fascinating, but it's also challenging. Because when I talk to biologists, they probably think I know nothing about biology, which is very true. So we are learning all the time. We are learning all the time, which is really great. But I think that the concept of combining pieces from different subjects should be introduced as early as possible. So we are actually very devoted to outreach activities, which I started to do in 2007. And since that time, we visited about 400 students from middle and high school. And you can see I think they have fun. So they're doing basic nanotechnology experiments, or we show them how those different pieces which they learn in biology and in sciences, they actually fit together. So this is probably the most important slide, showing my group. I have currently 7 graduate students, and I've talked about some of the work which was done by my alumni, who are listed on the right-hand side. So I really do not have any conclusions, other than we have a lot of fun with making holes. And actually, I talked to John, and he said, "Why did you enter the field of holes?" And maybe it sounds cheesy, I don't know, but I was a very weird child who started to talk at the age of 4. And that was a huge disappointment for my mom because I never liked dolls. Okay, so the dolls, so they were really worried about me in many ways. But I loved being in the basement with my dad. And of course, he didn't let me play with many gadgets because it was too dangerous. But at the age of 6 or 7, he gave me a hand drill, and he gave me a wooden block, and I could make little holes in the. And I thought that's really cool. Yeah, I was a really strange child. So, anyways, I think we are doing that still as we speak about. So I'd like to thank everyone who has been extremely supportive, and the list is much longer than it's here. And many thanks to Tatiana and Audrey for their amazing patience in organizing it and their help in preparing the abstract. And I would like to thank all the sponsors for money, and you for your attention. I will be happy to answer questions.
Okay, so 10 years from now, when UC Irvine is the home of squishiness science, you can say you heard it first. Any questions for, thank you for a wonderful lecture.
Thank you, Ken.
Any questions for Professor Siwy?
So the question is about that microstructures are very often clogged. And how do we deal with that problem? So surprisingly, at the nanoscale, we actually have less of an issue with that. And the reason for it is that the majority of stuff which happens to be in the solution is much larger than nano. So it simply doesn't really block it that well. Now on the microscale, it is a big problem. Okay, so on the microscale, especially with the cancer project, we have to make sure that we passivate the surface so that the cells will not like sticking there. Another way of mitigating that, so it's not an issue. But on the nanoscale, surprisingly, is extremely robust. It's very weird.
If I understand this, you said that the malaria cells are hard, so they're not squishy. I was wondering, has anybody done a correlation study between malaria and cancer?
I'm not aware of that study. Maybe there is. I mean, so one thing which I want to clarify is that the malaria is the parasite, which is mostly attacking red blood cells, is making the red blood cells much more stiff, much less squishy. If there is a correlation, that would be an interesting question, but I'm not aware if anyone is right now pursuing that line of research. But I mean, squishiness is very, I'm trying to avoid the word hot. It's very much research right now because people found out that if you put a cell on a surface, the cell actually tries to feel the surface, and it exerts the force on it, like pokes on it. So it's also thought that cells can communicate with each other by exerting forces on each other. So much more is known about chemical ways of communicating between cells, but very little is known about physical properties of these, and they are very important. Okay, so the question is you know what? You can do that. Oh, no, okay, I'm sorry. So the question was about desalination, and please correct me if I misinterpret your question. It's about desalination. What would be the next step of making it more viable, right? How to bring the technology to the level which would be useful for drinking water on an everyday basis. So there are different approaches, in fact, which people try to pursue. Our approach is based on the fact that, so basically this is so inefficient because the pores which you have in the current desalination units are really, really tiny. And the flux depends on the diameter to the 4th power. So if you make the pore really tiny, the flux will just drop tremendously, right? So one way is to think about another way of separating salt from water than the size of it, and then you could afford having a much larger pore. So even if you went up with the size to 2 or 3 nanometers, you would solve the problem. But now we have to think about how to make the how to make the separation feasible, right? Because really to make the water usable, we have to reject 99% of the salt, something which I did not appreciate at all. So it's a lot of salt which needs to be rejected. Another approach is to use structures which are called carbon nanotubes, which are very, very tiny, but they have this amazing property of transporting water as fast as aquaporin. So there are different technologies which are run in parallel, and people are working hard on making it realize. But it's clear that the heart of the desalination, which is the pores, has to be redesigned. There's no way around it.
Does your lab make measurements on both the biological systems and make your own pores? Or do you use the biologist interpretation and then make your own pores to match?
So the question is whether our lab is using both biological pores and our synthetic pores, or only one of them. So currently at UC Irvine, we work only with the synthetic man-made pores in the project, but we are collaborating with Professor Hughes from the School of Biological Sciences on the cancer project, because we really do not have, as of now, facilities to grow the cells. The cells are very picky. They get upset very easily, and I guess we make them upset. So they are growing them, and then we bring them on ice and study them in our eyes, which is okay for a few hours. So we really need to talk to biologists.
So I have one quick question. When they built the heavy ion accelerator in Germany, was there any hope? Did they even think about the possibility that it would lead to your work in making liquid-like electronic circuits?
I'm not sure they did, but but actually the accelerator, which I wanted to mention, is using maybe even for a grander purpose, okay? So the accelerator, when I was in Germany, oh boy, 14 years ago, the accelerator which we were using was shared with a medical group. And of course they had priorities, because heavy ion is used now routinely to cure brain cancer. So the reason for that is, so now they have a special accelerator in Heidelberg, so we do not have to share, or they don't have to share that with us. I guess that's the proper way of saying it. The reason for it is that there's no side effects, absolutely nothing whatsoever. So a person is irradiated with a really heavy energetic beam of carbon ions. And the beauty of it is that when it passes through healthy tissue, there is very little deposition of energy. So there's very little damage. So what they do, they tune the energy in such a way that the whole deposition, the dumping of energy, sorry for the bad word, occurs at the point of the cancer. So when the heavy ion passes in, it deposits very little energy, and there's a huge spike of the energy at the very, very end. And that happens at the cancer location. So it's in combination with CT, of course. They have beautiful, so they have been doing that for 15 years at least.
Okay, let's thank Professor Siwy for a marvelous lecture.
Thank you, Ken.